we're going to continue with the kidneys, and we had mentioned how filtration occurs, right? And it occurs in the renal corpuscle, which is the Bowman's capsule and the glomerulus. So that's the only location where filtration happens. Remember, the driving force for filtration is going to be that capillary pressure, which correlates with the blood pressure. It's going to depend, it's a hydrostatic pressure. It depends on how much volume of blood we have in that glomerulus. So we can regulate the filtration rate by affecting the radius of the afferent and efferent arterioles. Now I'm going to talk about the processes of reabsorption and secretion that occur in the rest of the nephron, and this is going to involve the other segments, the proximal tubule, the loop of Henle, distal tubule and collecting duct, and its association with peritubular capillaries. Okay? Now, the peritubular capillaries, they're just called that, peritubular capillaries. You see all these is going to be peritubular capillaries. However, these peritubular capillary here associated with the loop of Henle, it's called the vasa recta. And I'll talk about the vasa recta in a little bit, in a few slides. Okay, so in a day, the filtration, the process of filtration is going to make 180 liters, okay, of filtrate. So 180 liters of filtrate get made every day. But of that 180 liters, only 1.5 liters are excreted. So excreted going into the ureter, into the bladder, and then the urethra, and out, okay? So most of the things that you filter are going to actually be reabsorbed and brought back into the body, into the circulatory system, okay? So that what we excrete is just minimum. Let's look at how reabsorption and secretion occur. So reabsorption, remember, is moving materials from the lumen of the nephron, so from the filtrate, which is considered outside of the body, back into the peritubular capillaries, which are part of the circulatory system. It's inside the body. And then secretion, it's going to be moving things that are in the blood, in those peritubular capillaries, and we want to get rid of them, so we secrete them into the lumen of the nephron, into that filtrate to be excreted. Okay, so we have those two processes. Let's talk about reabsorption first. So reabsorption, most of the reabsorption happens in the proximal tubule. So proximal convoluted tubule, which is that first segment of the nephron after the Bowman's capsule. Okay, so first segment of the renal tubules. And about 70% of the materials are reabsorbed in these, or 70% of reabsorption, happens in this proximal tubule. And we're going to reabsorb a number of different things. We're going to reabsorb water, so about 50% of the water gets, 70% of the water gets reabsorbed. 70% of salts get reabsorbed. 100% glucose gets reabsorbed. About 50% urea gets reabsorbed in this first segment of the proximal tubule. So this proximal tubule is where most reabsorption happens. You're going to have reabsorption throughout the whole rest of the nephron, but 70% of the reabsorption happens in this initial segment of the renal tubule. All right. How does reabsorption happen in the proximal tubule? It happens using sodium as the driving force. So remember that we have a sodium-potassium ATP pump, and we always have a sodium-potassium ATP pump in all the cells. These walls of the proximal tubule are made of proximal tubule cells, right? So the way it would look like is if you, let me get my pen here, if you were to look at that proximal tubule, you have your proximal tubule here. Here's the Bowman's capsule. This is made of cells, proximal tubule cells. And you always have two membranes in those cells, in cells. You have the basolateral membrane, okay? And we call this the apical membrane. The apical membrane always faces the lumen, in this case the lumen of that nephron, so it's going to be in contact with the filtrate. The basolateral membrane is the same cell, but we give different numbers to the cell because we have different transporters in the different parts of the membrane. The basolateral membrane, which is going to be facing the extracellular compartment, is going to be facing those peritubular capillaries, right? Here and here. These are the peritubular capillaries. That basolateral membrane always has a sodium-potassium ATP pump. So I'm going to write the name here, sodium-potassium ATP pump. And you guys study those pumps already, right? What the sodium-potassium ATP pump does is move potassium into the cell and sodium out of the cell, creating a concentration gradient for these ions, right? So you're always going to have more sodium on the outside than the inside, right? You're going to have more sodium on the outside than on the inside because these sodium-potassium ATP pumps are going to move sodium out of the cell. So you have a driving force for sodium. So sodium is going to move down its own concentration gradient, that's the driving force, given that we have a pathway for sodium. And we have a pathway for sodium because we have a number of sodium channels, like epithelial sodium channels, symporters that transport sodium as well, and antiporters. So sodium is going to be able to move from the filtrate into the proximal tubule cells and out using its, here. Let me draw it a little better. Yeah, so sodium is going to be able to move from the filtrate down its concentration gradient, right? Because you have very little sodium here in the cells, right? So then sodium is going to move down its concentration gradient from the filtrate into the cell, and then you have on that basolateral membrane sodium-potassium ATP pumps that move it back out. And let me draw it here. So sodium is going to move here into the cell and then back out by the sodium-potassium ATP pump. So you see that sodium over here, it's moving, it's being reabsorbed. It's going to end up in these peritubular capillaries. All right, using its concentration gradient, and we have those epithelial sodium channels that allow sodium to move through. So those channels are going to be here. Now, it allows us to move other solutes. So sodium is going to be that friend that gives you a free ride, right? So sodium is indirectly using ATP to create that concentration gradient. We have that sodium-potassium ATP pump using ATP so that the concentration of sodium outside the cell is higher, and that allows us now to move sodium from the filtrate into that cell, and then the sodium-potassium ATP pump keeps moving it out, and then it goes into the peritubular capillaries. And this is through sodium channels, right? Sodium channels located on the apical membrane, and the sodium-potassium ATP pump on the basolateral membrane. So we have a pathway, we have a driving force. Now, there are other transporters that are going to allow glucose and amino acids move from the filtrate, the lumen of that nephron, into the cell and out into the interstitial compartment and then the extracellular compartment, the peritubular capillaries, the blood in the peritubular capillaries, using transporters like, for example, sodium glucose transporter, amino acid transporters that work with sodium as well. So we're using secondary active transport. Secondary active transport is when we don't directly use ATP, for example, to move glucose through a sodium glucose transporter. We're using the driving force for sodium, but indirectly use ATP by that sodium-potassium ATP pump creating that concentration gradient. So the way it works is you're going to have, and let me change my ink color right here. You're going to have sodium glucose transporters that over here, right? This is going to be a sodium, and I'm just going to glucose transporter. I'm just going to write it like that, sodium glucose transporter. That it's going to move glucose from the filtrate into the cell against this concentration gradient, because remember, we always have more glucose inside the cells. You should have learned that in Build 1, right? There are always more glucose inside the cell than outside the cell. So we're moving glucose against this concentration gradient without using directly ATP, because sodium is helping us move using its concentration gradient. That concentration gradient created by the sodium-potassium ATP pump right here. Does that make sense? So now glucose and sodium move into that proximal tubule cell. Sodium-potassium ATP pump is going to be in charge of moving sodium again outside. And glucose now moves through simple diffusion, through just a regular glucose transporter, because we have too much glucose now inside the cell. It has a driving force, and we happen to have a pathway as well. Does that make sense? And that's how amino acids and other solutes get moved, getting the help from sodium, secondary active transport. Does that make sense, guys, or no? Yep. So we are going to absorb a number of nutrients like amino acid and glucose using the driving force for sodium. Any questions about that? Okay. Now, water will follow due to the osmotic pressure generated by the movement of the solute. So if we reabsorb sodium, glucose, amino acids, bicarbonate is another one that's a different pathway. All these different solutes are going to move from the filtrate into the interstitial compartment. So this is the interstitial compartment right here, and then into the blood in the peritubular capillaries. So you're going to have peritubular capillaries right here and peritubular capillaries right here. So that's going to make it so that the solutes now in the peritubular capillaries generate a higher osmotic pressure than in the filtrate, because the solutes move. Remember what osmolarity is. Osmolarity is the number of solute particles per volume. Right? So if a bunch of those solutes move now out of the filtrate into the blood in the peritubular capillaries, the osmolarity in that filtrate is low relative to the osmolarity in the blood. Proximal tubule cells are permeable to water, so water will follow, right, to try to equilibrate that higher osmolarity now in the peritubular capillaries. And as water follows, then we're going to start having a higher concentration of urea in the filtrate, in the lumen of that nephron relative to the blood in the peritubular capillaries. So some of that urea is going to move through urea transporters back outside of the nephron from the lumen of the nephron into the interstitial compartment. That is important, and I'll explain why it's important a little later. But urea, we don't want to have too much urea in the blood because urea denatures proteins. So some of it will move into the blood, but most of it will stay in the interstitial compartment, the compartment between cells, the compartment between those proximal tubule cells that make the proximal tubule wall and the wall of the peritubular capillaries. Okay? And in keeping some of the urea there, we're going to maintain the osmolarity of that interstitial compartment of the renal cortex. Okay? Remember I told you that the kidneys are divided in renal cortex, which is the outside, renal medulla, which is the inside, and some of the nephron is going to be in the renal cortex, some of the nephron is going to be in the medulla. So the Bowman's capsule and the proximal tubule are going to be in the renal cortex, as you see right here. So right here, you see this is all the cortex. The proximal tubule is in that renal cortex. Any questions about this? Okay. Now secretion. We're going to secrete in the proximal tubule things like hydrogen ions to regulate pH. So we reabsorb bicarbonate, bring it back into the blood. So from the filtrate in the lumen of that proximal tubule into the blood in the peritubular capillaries, and secrete hydrogen ions. So from the blood into the filtrate to be excreted. This is to regulate pH. And then some pathogens as well that we want to get rid of. Some drugs as well will be secreted into that filtrate in the proximal tubule. So because water, even though all these solutes were reabsorbed, right? Glucose, amino acids, salts being like chloride, sodium, potassium, all these things got reabsorbed in the proximal tubule, got moved from the filtrate in the lumen of that proximal tubule into the blood in the peritubular capillaries. The plasma of the blood, the plasma of the renal cortex, and the plasma of the blood, the oh my God, I need that coffee. The osmolarity of the blood, of plasma in the peritubular capillaries, the osmolarity in the renal cortex, and the osmolarity in the filtrate of that proximal tubule are going to be isosmotic. They're all going to be 300 milliosmolar. Why? Because even though we move all those solutes into the blood, the water moved as well, right? And now we have equal osmolarity. The water moved to dilute those solute particles in the blood, and so we have the same osmolarity in all the three different compartments. All right, so plasma, the renal cortex interstitium or interstitial fluid in the renal cortex, and the filtrate osmolarity are all going to be 300 milliosmolar. So isosmotic, and that's important, and I'll explain why later on, or it'll become clear later on. All right. So what happens after the proximal tubule? After the proximal tubule, we have the loop of Henle. So remember, I told you need to know about the descending limb and the ascending limb of the loop of Henle. Okay? The renal cortex, as I already told you, has an osmolarity of 300 milliosmolar. The filtrate in the proximal tubule has an osmolarity right here of 300 milliosmolar. And here is not shown, but we should have a proximal tubule is associated with the peritubular capillary. It's not shown here. But the peritubular capillary will have that blood with the same osmolarity, 300 milliosmolar. As we move down into the renal medulla, so you see here is the renal medulla. The renal medulla has an osmotic gradient. It's not going to be 300 milliosmolar. The renal medulla goes from 300 milliosmolar all the way in a human to 1200 milliosmolar. This renal medulla osmolarity varies between different species. So pigs don't have a deep renal medulla, so they have lower osmolarity. Kangaroo rats that live in deserts, you guys probably have heard of them or seen them in Ansa Borrego here. They have a very deep renal medulla, and the renal medulla goes up to 9,000 milliosmolar. This is important because it allows the animal to conserve water. So the deeper the renal medulla and the higher the osmotic gradient, the more water conservation we can have. Whales also have a very deep renal medulla, okay? And I'll explain why, or it'll become clear at the end of this lecture. So important thing right now, I want you guys to know that there is an osmotic gradient in the renal medulla. The renal cortex is all the interstitium, or interstitial fluid of the renal cortex is all 300 milliosmolar. But the renal medulla interstitium, or medullary interstitium, is going to have an osmotic gradient. In humans, it goes to 1200 milliosmolar, okay? Now, when the filtrate gets here at the end of the proximal tubule, the osmolarity is 300 milliosmolar. So it's isosmotic to plasma, it's isosmotic to the renal cortex interstitium. As it moves in the descending limb of the loop of Henle, This epithelium, the cells that make the walls of the descending limb of the loop of Henle, are permeable to water, but they're not permeable to any solutes, only to water, okay? So these cells that make the wall of the descending limb of the loop of Henle only have aquaporins, channels for water, okay? And it doesn't allow any solutes, no sodium, not glucose, not hydrogen ions, nothing else can move, only water. So we have pathway only for water. Now, at this point, you see that as the filtrate is going down deep in the renal medulla, water is going to be moving because we have a pathway. My question to you guys is, do we have a driving force? What is the driving force for water to move? I told you we have water channels called aquaporins. So this is permeable to water, the descending limb of the loop of Henle. What would be the driving force, the gradient? So we start with a filtrate here that is 300 milliosmolar. What would be the driving force? Anybody? Yeah. The osmotic gradient. The osmotic gradient. So you see that as you go deep in the renal medulla, this interstitium, the interstitial fluid, remember the fluid that is in the interstitial compartment, the compartment between cells. What cells? These cells of the descending limb of the loop of Henle that make the wall of the descending limb of the loop of Henle. And we're going to have a peritubular capillary here called the vasa recta. Okay? It's not shown here, but you have a peritubular capillary associated with all the segments of the nephron. So in the renal medulla interstitium, we have an osmotic gradient going from 300 milliosmolar up here all the way to 1200 milliosmolar. So as the fluid is moving down in the descending limb of the loop of Henle, and I'm getting my pen right here to draw. This is 300, right? And that 300 filtrate is going to encounter a 400 osmolarity. So that's a driving force. That's a difference in osmolarity, right? This is hyperosmotic relative to the 300 here. So water will want to move to where we have higher solutes, lower water, right? So water moves and we have a pathway. We have those aquaporins, okay? And I'm going to write here aquaporins so you guys know. Those are water channels, okay? Aqua means water, porins means pores. Then that filtrate lost a bunch of water, right? So we have more solute particles per volume. So now the osmolarity of the filtrate is 400 milliosmolar. But now here it's 500 milliosmolar. So again, we have an osmotic gradient. So more water leaves the lumen of that descending limb of the loop of Henle. Now we have here a filtrate that is 500 milliosmolar. But now we encounter here a 600 milliosmolar interstitium. So again, we have a difference, right? The renal medulla is hyperosmotic, so then more water leaves, and we keep losing water until it equilibrates at the end, at the bottom of the descending limb of the loop of Henle, it equilibrates with the bottom osmolarity of the renal medulla. So we have a very concentrated filtrate at the bottom of the loop of Henle. Does that make sense? Why? So we have the pathway, it's the aquaporins. We have the driving force, which is the osmotic gradient. So the filtrate, in this case, it's going to be at the bottom of the loop of Henle. The filtrate is concentrated. So the filtrate is going to be what? Iso, hypo, or hyperosmotic to plasma. What do you guys think? What is the osmolarity of plasma in a regular capillary? What is that number that I told you need to know? 300 milliosmolar. So if plasma were to be 300 milliosmolar. And the filtrate at the bottom of the loop of Henle is 1200 milliosmolar. The filtrate is going to be what? Iso, hypo, or hyperosmotic to plasma. What's that? Hyperosmotic, right? Because the filtrate is 1200 milliosmolar and plasma 300 milliosmolar. Now in the vasa recta, that is going to be a little different, and we'll talk about it in a minute, but the plasma in most of the rest of the nephron, it's going to be 300 on the peritubular capillaries, it's going to be 300 milliosmolar. So filtrate at the bottom of the loop of Henle is going to be hyperosmotic to plasma. All right. Now let's look at the ascending limb of the loop of Henle. So what happens now when we ascend here? Ascending limb of the loop of Henle is not permeable to water. Not permeable to water. So water cannot move. Even though we will have a driving force, right? The osmotic gradient is decreasing. We do not have a pathway for water, so water cannot move. But in that ascending limb of the loop of Henle, these cells that make the walls of the ascending limb of the loop of Henle have transporters for sodium, for solutes. So the different transporters are going to be, and I'm going to write them here. You have transporters for sodium chloride, so you're going to have sodium-potassium ATP pumps. You're going to have another type of transporter called the NKCC, which the NKCC is sodium, potassium, chloride cotransporter. And that's going to help with the driving force for sodium created by the sodium-potassium ATP pump. Sodium is going to move now out of the filtrate into the peritubular capillary here, the vasa recta, and it's going to help 1 potassium and 2 chlorides to move. Okay? And so we reabsorb all of these solutes. Okay? And we have urea transporters. So other transporters are going to be urea transporters. And that urea transporter is going to maintain this osmolarity. So some of these solutes are going to move into the peritubular capillary that is not shown here, but the vasa recta, and some of the solutes are going to remain, mainly urea, to maintain this osmotic gradient in the renal medulla. Okay, so what did we just do? We maintained the osmotic gradient by some of these solutes are going to move into the peritubular capillary, but some of these, so here is going to be the peritubular capillary, the vasa recta. So some of those solutes are going to move into the blood, but some of the solutes, like urea, some sodium, are going to remain in the medulla, the renal medulla, maintaining this osmotic gradient. So that's one of the roles of the loop of Henle, maintain the osmotic gradient in the renal medulla. The second role of the loop of Henle is to make a very dilute filtrate at the end of the loop of Henle. So a hyposmotic filtrate. Okay, look, the osmolarity is 100 milliosmolar at the renal medulla. Sorry, the renal cortex, we have a 300 milliosmolar. Plasma is 300 milliosmolar. So we're making a hyposmotic filtrate, a dilute filtrate. What happens next? So then we have, we enter the distal tubule and then the collecting duct. And the distal tubule and collecting duct they make the distal nephron. And we give it a different name to the distal nephron because reabsorption of water and solutes in the distal nephron is going to be under hormonal regulation. Okay? So it's going to be regulated. Water in the proximal tubule and descending limb of the loop of Henle was done due to an osmotic gradient and a pathway. reabsorption of solutes here, and in the proximal tubule, again, was with the sodium gradient created by the sodium-potassium ATP pump. What happens in the distal tubule and the collecting duct is going to be regulated, homeostatically regulated, okay? And it's under hormonal control. So let's look at what happens here. If we want to regulate osmolarity, okay? So here I have normal osmolarity of the blood. It's around 300 milliosmolar, so it goes between 275 to 195 milliosmolar. Just make it average 300. I know average is not going to be 300 milliosmolar here, but just to make it easier, say 300 milliosmolar. So we have the blood at 300 milliosmolar. If the blood osmolarity increases, so for example, if you're severely dehydrated or you were bleeding out and you lost a lot of blood, remember plasma is 93% water, or you're perfusively sweating, but I mean not a little bit sweating, you're perfusively sweating, right? Or you're going in heat shock or something like that, that is going to increase the osmolarity of the blood. Why? Because remember what osmolarity is the number of solute particles per volume. So osmolarity number of solute particles per volume. And if you're severely dehydrated or you're sweating too much, losing too much water, right, or you're bleeding out, this volume will go down. And if this volume goes down and the solute particles remain the same, then osmolarity is going to go up. It's going to go higher than 300 milliosmolar. Okay? So that osmolarity of plasma is going to be sensed by osmoreceptors. Osmoreceptors are the receptors or sensors that are going to monitor that osmolarity. You have osmoreceptor in different parts of the body, but the main ones are going to be in the hypothalamus in your brain. Okay? So the hypothalamus is going to get that information. Osmoreceptors in the hypothalamus sense that high osmolarity of body fluids and sends a signal to the hypothalamus. So the hypothalamus is also the integrating center. So it's going to integrate that information compared to a set point. The set point is 300 milliosmolar, right? And because osmolarity is higher than 300 milliosmolar, we are going to send an output signal to reverse the change in that physiological variable, reverse the change in that osmolarity, bring osmolarity back down. So what do? We have the hypothalamus send a signal to the posterior pituitary gland, and the posterior pituitary gland, remember, it's a continuation of the hypothalamus that stores the hormones made by the hypothalamus, oxytocin and antidiuretic hormone. So here, antidiuretic hormone, or vasopressin is the other name, gets released by the posterior pituitary gland into the circulatory system. It's a hormone, okay? Now, that hormone travels throughout the whole body, but in the collecting duct and in the distal tubule, which we call together the distal nephron, we have receptors for the antidiuretic hormone, okay? In this case, then, we are going to send the signal for water reabsorption. We're going to insert a pathway that wasn't there before, and we have the driving force, and I'll explain what the driving force is in a minute. If we reabsorb water, basically we're moving it back from the filtrate into the blood, right? We're reabsorbing water. We're going to increase this again, right? So we're going to now correct for that. We're reabsorbing water, meaning we're moving it to the peritubular capillaries. The peritubular water is moving into the peritubular capillaries. By doing that, we increase volume, right? We're bringing that blood to the that water to the plasma. We increase the volume, and so in this case, we decrease the osmolarity again. We bring it back to 300 milliosmolar, to homeostatic levels. The other thing that happens is that when the hypothalamus gets that we're hyperosmotic. The osmolarity of body fluids is too high. Hypothalamus is the homeostatic control center. So it's going to control certain behaviors like feeling thirsty, and then you're going to drink a bunch of water, and that replaces some of the fluid that you lost. So this is a behavioral regulatory mechanism. So you have the physiological and you have the behavioral. All right, let's look at how antidiuretic hormone causes water reabsorption. Well, here I'm showing you the wall of this distal tubule. Okay, remember we have all these made of cells, and of the collecting duct. Okay, so what I'm showing you here, these cells right here, okay, are these cells right here. All right, so these are the different cells that make the walls of the distal tubule and the collecting duct, the distal tubule and collecting duct. All right, and here you have the lumen of that nephron that has the filtrate. It's shown yellow to show the filtrate. Here you have the interstitial compartment. So if you are looking at the collecting duct, this is going to be renal medulla interstitium. If you're looking at the distal tubule, then this is going to be renal cortex interstitium. Okay? And what the antidiuretic hormone is doing is it's going to be, remember you have your peritubular capillaries right here. You have your peritubular capillaries right here. Okay? So you're going to have peritubular capillaries right here and right here. And so that's with blood, right? So this has blood in here. So antidiuretic hormone, remember, is released by the posterior pituitary gland. It's going to travel all throughout the circulatory system. When it gets to the distal tubule and collecting duct, it's going to bind to an antidiuretic hormone or vasopressin receptor that is located in the membrane of the membrane of the cells. Remember, this antidiuretic hormone has to bind to a membrane receptor. It cannot cross the cell membrane because it's hydrophilic or lipophobic. Okay, so it activates this receptor. It binds to the receptor and initiates a signal transduction cascade that ends up producing cyclic AMP. You learned this in Human Physiology 1, the specific pathway. For here, you just know that cyclic AMP, which is a secondary messenger, is produced, and that is going to cause activation of a protein kinase, specifically protein kinase A. What does a kinase do? A kinase phosphorylates. So it's going to phosphorylate these storage vesicles. These storage vesicles, what they are storing is aquaporins. It's really cool. This is called membrane recycling. So when they get phosphorylated, these storage vesicles go and fuse with the apical membrane of the proximal tubule and distal tubule cells. So this is the apical membrane, because it's the one facing you should know these names, is the one facing the lumen, the filtrate. This is the basolateral membrane. They fuse with the apical membrane and they insert the aquaporins. So now water has a pathway. It didn't have it before. So you need that antidiuretic hormone to insert this pathway for water. So water can move into the cell, and you always have these aquaporins on the basolateral membrane. Okay, so these aquaporins here actually they're a different isoform. They're aquaporin 1s. And they're always present in the basolateral membranes, always there. So these ones are always there in the basolateral membrane. But the apical membrane has aquaporin 2s. And so aquaporin 2s are only inserted on the apical membrane if we have a signal from that hormone, antidiuretic hormone or vasopressin. So what happens here is that if antidiuretic hormone or vasopressin is present, then we're going to insert a pathway for water. Water is going to be able to move into the peritubular capillaries, and you're going to end up excreting a very concentrated urine. So you know when you're dehydrated, and the code is 45, guys. When you're dehydrated, the urine comes out dark, like a dark yellow, right? That is because you're excreting solutes with very little water. You reabsorb the water to bring it back into your body. If you have no vasopressin present, all right, if the signal for vasopressin release does not happen, you have no vasopressin, then you do not have a pathway for water. Even though you have the driving force, you don't have a pathway for water. Water doesn't move, and you produce a diluted urine. Okay? Now let me ask you a question here. How do you, you have the pathway, I'm telling you about the pathway here, inserting those aquaporin tubes under the signal of antidiuretic hormone, right? What is the driving force for water reabsorption in the collecting duct? So what is giving me this driving force? Look at this. The osmotic gradient, right? So in the collecting duct, we're good because if we have the driving force always there, we have that osmotic gradient, right? And what we're inserting now with ADH is the pathway. How about the renal cortex? Because the renal cortex doesn't have an osmotic gradient. It's always 300 milliosmolar. What is the driving force for water reabsorption? And do we have a driving force? Do we have a difference in the osmolarity of the filtrate in the distal tubule and the osmolarity of the cortex interstitium, renal cortex interstitium? What is the filtrate osmolarity at the distal tubule? At the distal tubule. Remember that distal tubule is right after the loop of Henle. Remember, one of the main roles of the loop of Henle was to produce a hyposmotic filtrate of 100 milliosmolar. So the distal tubule, which is this whole thing, has a filtrate of 100 milliosmolar. And the renal cortex is 300 milliosmolar. Do I have a driving force? Do I have a gradient? I do, right? And this is higher than this. So will I have water move out of the filtrate? We do, right? We have a driving force. It's key that we make a filtrate that is hyposmotic at the end of the loop of Henle, because that will make it so that I have the driving force for water reabsorption if I have the pathway of those aquaporin tubes inserted due to antidiuretic hormone signal. Does that make sense? Because here I have no problem. I always have that osmotic gradient that is going to give me that driving force. But here, this filtrate needs to be hyposmotic so that I can, to the renal cortex interstitium, so that I can have that driving force. All right, lastly, I want to talk about regulation of pH. And remember I told you that the lungs are the ones that take care of about 70% of pH regulation by either increasing breathing or decreasing breathing, because that's going to affect the amount of CO2 we have in our blood, and that is going to either move that equation to the left or the right, producing more hydrogen ions, or decreasing the concentration of hydrogen ions, therefore affecting pH. So we learned that with the respiratory system. The other system that can regulate pH is going to be the kidneys. So they can reabsorb or secrete bicarbonate and secrete or reabsorb hydrogen ions. And this happens in the proximal tubule and in the collecting duct. Those are the places where we have pH regulation. So let me ask you a question. If bicarbonate is reabsorbed into the peritubular capillary, so brought back into the body, into the circulatory system, how will blood pH be affected? Will blood pH increase, decrease, or not change? So you're moving bicarbonate into the circulatory system, into the blood. Is bicarbonate going to increase, decrease, or not change pH of the blood? Remember what bicarbonate does. Bicarbonate reacts with hydrogen ions, forming carbonic acid. So it's going to buffer these hydrogen ions. Once we bind that hydrogen ion to the bicarbonate forming carbonic acid, that hydrogen ion concentration will no longer contribute to pH, right? So what do you think would happen? If we're decreasing the amount of hydrogen ions because it's binding to bicarbonate, because we're increasing bicarbonate, what do you think will happen to the pH of the blood? Increase, right? pH increases, or it's becoming alkalotic. So the law of equilibrium dictates the direction of this equation, right? If blood bicarbonate increases, the concentration of hydrogen ions is going to decrease because you're going to have bicarbonate and hydrogen ions react, forming carbonic acid, okay? And if the concentration of the hydrogen ions decreases, this is going to cause an increase in pH, all right? Meaning alkalosis, okay? So here we have bicarbonate increasing is going to decrease because this equation is now going to go in this direction, forming carbonic acid, then dissociated into water and CO2. Now, what would happen if bicarbonate is secreted into the filtrate state instead? What would happen to the blood? If now, this is if we're reabsorbing bicarbonate, bringing it back to the blood, right? What if now, instead of reabsorbing bicarbonate, we're secreting it, we're putting it into the filtrate to be excreted? What would happen to the pH of the blood? The opposite, right? It will decrease. If now we are decreasing this in the blood, right? That means that we're going to have the equation go in this direction, and less of it is going to, less bicarbonate reacting with the hydrogen ions. You're going to have a higher concentration of hydrogen ions in the blood. The pH of the blood decreases. It's acidotic. All right. It's time for you guys to go. I'll finish up with this regulation, and then we'll do digestion next week.